Okay, so we are done with the basic structure of the DNA. Right now, the motive for this particular video is the packaging of the DNA hex. So, we have got 23 pairs of DNA, right? So we have got 23 pair of DNA. 23 pairs of DNA, the molecules of DNA, and one molecule is known as one particular chromosome. So we have got 23 pair of chromosome. Okay, so if you are going to take your chromosome number 1, and from this, if you're going to calculate that after this is the chromosome number, let's say 23, up until there, so this is somewhere around 1 meter of the DNA. This is like somewhere around 1 meter, and since you have got 2 copies, 23 pair, then this will be the next another 1 meter of the DNA. So each cell inside a body contains somewhere around 2 meter of the DNA. And you can see that this is 2 meter, which is supposed to be packed into the cell, which is on to the level of micrometer. So it needs to be packaged well, so that it can be placed inside the cell. So how it is getting packaged? So number one thing, that our DNA is because of phosphate, it is very much negative. Okay, what is it? Negative. So because of phosphate, it is negative. What can be done? If you can have some protein, okay, you can have some protein which are positively charged. Protein number 1, protein number 2, protein number 3. So what can happen? This is your protein. DNA can be wrapped around this particular protein. So this is how it is being wrapped. One round, this is next round. So this is how it can be packaged. Okay. So it is being packed. Positively charged protein, positive charged protein, and negatively charged DNA. So it is going to wrap around it. Let us try to find out what this protein is. This protein is known as histone protein. Now histone protein can be divided into different subunits. Okay, what different subunits? These units are number 1 H2A, number 2 H2B, H3 and H4, and you must be asking where is H1. So okay, H1 as well. So H1. So these are the different units. So you can say different polypeptides. Okay, so this is one polypeptide. This is one polypeptide. So these are different protein which come under one particular category of histone proteins. So now what is going to happen, that 2 DNA A, okay, so DNA A, DNA B, DNA H3, H4. Again one more, H2A, H2B, H3, H4. We have got 1 2 3 4 5 6 7 8 protein. Okay, so what they were H2A, H2B, H2A, H2B, H3, H3, H4, H4. Okay, so this is how we have got 8 molecules of protein, and these makes one. So this will make a one big structure, and this big structure, as you can see right here, so this is that big structure, and around it we have packaged our DNA like this. Okay, so this is how the DNA is being packaged around one particular protein, which is known as the histone. Okay, let me take the highlighter. So this is histone protein, and histone protein are known as histone octamer. For how is it making the histone octamer, you just got to know H2, H2B, H3, H4 and H1. So right now, can you see this particular histone? This particular histone is known as H1. Why? Because if there are 2 histone along with their DNA, okay, histone along with their DNA, if there are 2, then the distance between them can be managed by or manipulated by this H1. Okay, the question comes that which of the following is not a part of histone octamer. So the histone octamer does not have H1, so this will be the answer. It is helping out to manipulate the distance between 2 histone along with their DNA, and this particular unit is known as the nucleosome. So histone along with the DNA around it is known as nucleosome. And one nucleosome has got somewhere around 200 base pairs around it. So this will be somewhere from start, if we start from right here to wherever it leaves the histone protein, there will be somewhere around 200 base pairs of. Okay, so this is how, and if you can see into this figure as well. So these are those nucleosomes, okay, and which looks like beads on string. These beads on a string can occur, okay, into two formats, okay, and this either they can be very much far apart from each other, or either they can be very much closer to each other. Okay, this structure, this is known as chromatin. What is it known as? Chromatin. Chromatin. And chromatin can be divided into two parts, two kind, how it can exist. One into the loosely packed form, which is known as your euchromatin, and this will be into a densely packed form, which is heterochromatin. Euchromatin, it is being loosely packed. Why? Because the DNA is very much ready to make the RNA. Okay, so DNA is very much ready to make the RNA. So you know this thing, that your pancreatic cell, if this is the pancreas actually makes, let's say, insulin, and your hair cell make keratin, let's say keratin. So pancreas is making insulin, hair cell is making keratin. Pancreas cannot make keratin and hair cell cannot make insulin. But in spite of this fact, this cell, the cell of the pancreas, has got gene for the DNA which can code for that particular protein. It has gene for keratin as well as for insulin. And the hair cell also have got DNA for both insulin as well as keratin. But hair cell never makes insulin, it always makes keratin. So how does it happen? So this actually depends at how the DNA is present inside that particular cell. So the insulin gene is present in somewhat euchromatin form into the pancreas, and the keratin gene is present inside the pancreas into the heterochromatin form. That is why it is not getting expressed. Okay, so this is the basic idea regarding the packaging of the DNA. Okay, so there are some proteins which are known as the non-histone chromosomal protein, which are present into prokaryotes, and they actually can package their DNA onto the non-histone chromosomal proteins. And euchromatin and heterochromatin we have already discussed. So we'll meet into the next video, in which we are going to discuss onto the history of the genetic material.